Hello, we'll start today's lecture about hematology. Hematology actually is a branch of science, part of a physiology, pathology, clinical laboratory, internal medicine that is concerned with the study of blood and the blood-forming organs and the blood diseases. It includes the study of etiology, diagnosis, treatment, prognosis, and prevention of blood diseases. Blood diseases that affect the production of blood and its components, such as blood cells, hemoglobin, blood proteins, and mechanism of coagulation. Some of the common basic hematology tests include full blood count, ESR, blood film, bone marrow examination, COMS tests, serum ferritin level, vitamin B12 and folate levels, prothrombin time, partial thromboplastin time, protein electrophoresis, and hemoglobin electrophoresis. The classification of hematological disorder, we have hemoglobinopathies, which is a congenital abnormality of the hemoglobin molecule or of the rate of hemoglobin synthesis. We have sickle cell disease, thalassemia, methemoglobinemia, anemias, myeloproliferative disorder, like means increased number of cells, as polycythemia vera, leukocytosis, thrombocytosis, and this is increased number of platelets. Hematological malignancies as lymphomas, coagulopathies in disorder of bleeding and coagulation, as hemophilia and thrombocytopenia. The blood is several types of cells, represent 45%, and the plasma 55%, while the hematocrit is the proportion of blood occupied by red blood cells. The normal pH of the blood is around 7.35 to 7.45. It's a weak alkaline solution. Below 7.35 is acidic, and the vice versa, and above is alkaline. The blood pH readings are very helpful in determining the acid-base balance of the body. The respiratory system and the urinary system normally control the acid-base balance as part of hemostasis. Blood is about 7% of human body weight, so the average adult has a blood volume of about 5 liters. The function of blood actually supply oxygen and nutrients to tissues, removal of waste, immunological functions and coagulation, messenger function, and regulation of body pH and body temperature. The composition of blood. Blood is divided into plasma and cells. The cells actually, it's erythrocytes, leukocytes, and the platelets, thrombocytes. The erythrocyte is the most common, second in size, early identified by their unique biconcave disc shape, formed in the red bone marrow, live about 120 days and removed by phagocytosis. Actually, the spleen is the primary site of phagocytosis of aged RBCs. The liver and bone marrow also play a role in removing or recycling RBCs. The main function, as you all know, of erythrocytes is carry oxygen to all parts of the body and bring back carbon dioxide to the lungs. The leukocytes is the largest in size, fewer number than red blood cells. Two major types of leukocytes, either granulocytes or agranulocytes. The granulocytes, which is a BMN, polymorphonuclear leukocytes, are the most numerous, common. There are darkly stained granules in the cytoplasm. These granulocytes are further classified into neutrophils, eosinophils, and basophils. The agranulocytes, which is the mononuclear leukocytes, they have no dark staining granules in the cytoplasm. Agranulocytes are further classified into lymphocytes or monocytes. Leukocytes are chiefly responsible for fighting infection by identifying, engulfing, and destroying foreign organisms. They are very active in the immune response by producing antibodies to foreign organisms. The thrombocytes, and the third type of cells, which is platelets, and they are fragments of megakaryocytes. They are essential for coagulation. Without an adequate number of these platelets, an individual would bleed to death. Blood collection procedures. There are two basic types of blood collection. There are the skin, dermal puncture, and venipuncture. We have a skin puncture is used to collect capillary blood. The puncture site for adults and children is usually from the fingertip, and for infants, the site are the heel or the great toe. The venipuncture is necessary for most diagnostic tests. Blood is drawn from a vein through the use of a needle and a syringe. The venipuncture in medicine, venipuncture or venipuncture, phlebotomy, venesection is the process of obtaining a sample of venous blood. Usually from 5 to 25 ml sample of blood is more than enough. After the needle is inserted into the vein, the phlebotomist receives the confirmation of success when a small amount of blood appears in the back of the needle, and this they call it a flash. The plunger is then pulled backward and blood fills the syringe. If the syringe plunger is pulled back too quickly, RBCs may be broken because of hemolysis by turbulence or physical force as they are forced through the needle. The blood is usually transferred quickly into a vacuum container tube. The blood cultures. Most often blood culture are drawn using a butterfly needle. When drawing blood cultures, the aerobic culture should be drawn first. Since there is air in the butterfly needle, tubing anaerobic specimens cannot be drawn without first removing the air from the tubing. So you need to first remove the air and then collect the blood afterward. The tops of any containers used when drawing a blood culture should be sterilized. Blood alcohol test. It's generally not advisable to use isopropyl alcohol to clean the venipuncture site or the site for obtaining blood. When obtaining specimen for blood alcohol test, to test the blood alcohol levels, so you need to clean with soap and hot water. This is an acceptable alternative to isopropyl alcohol or ethanol when collecting blood sample for back tests. Some important key terms. Anticoagulants, of course, it's the agent that prevent blood clotting like ETTA, citrate, or heparin. Capillary, actually, it's a small blood vessel that connects the arterioles with venules. Hematomas, a subcutaneous mass of blood at the site of the collecting the blood. Hemoglobin, it's the oxygen-carrying molecule of the red blood cells. Hemolysis is the breakdown of red blood cells with the release of hemoglobin into the plasma or serum. We cannot use the hemolyzed sample in lab tests. Isoteric is a jaundiced dark yellow or greenish serum or plasma. Lipemic, having abnormal high level of fat and the samples look milky. Plasma, is the difference between plasma and serum. The plasma actually is a pale yellow part of the whole blood, contain all the clotting factors, while the serum, it is a liquid portion of blood without any protein of fibrinogen responsible for clotting, which one of the clotting factors. That means we remove the clot. Agglutination, it is the clumping together of RBCs by the action of an antibody. When A cells are added to a blood sample, agglutination will occur only in the presence of anti-A. Serology, it is the study of antibody reaction using laboratory tests. CBC, complete blood count, is also known as full blood count or full blood exam or hemogram. Abnormally high or low counts may indicate the presence of many forms of disease. Three major variables in whole blood: amount of hemoglobin present, hematocrit or proportion of red cells, absolute number of red cells. A complete blood count will normally include all of these parameters, which we're going to talk in details in the next slides. Purposes of CBC. CBC, it's a pre-operative test to ensure adequate oxygen carrying capacity and hemostasis. Identify person who may have an infection, diagnose anemia, identify acute and chronic aliens, bleeding tendency, and white blood cell disorders such as leukemia. Monitor treatment for anemia and other blood diseases, and determine the effects of chemotherapy and radiation therapy in blood cell production. The precaution. The CBC requires a sample of blood collected from a vein. The tourniquet should be removed from the arm as soon as the blood flows. If a finger stick is used, care must be taken to discard the first drop and not to squeeze the finger excessively. Many drugs can affect the result by causing increase or decreases cells or platelets production. Medication as well should be taken into account when interpreting results. The samples. The blood is drawn in a test tube containing an anticoagulant. Counting the cells in a patient blood may be performed manually or automatically by use of automated analyzer. Automated counting is applied using a cell counting machine counters. These are quick, precise, quantitative analysis and give you the number of everything, including the blood cells, RBCs, indices, amount of hemoglobin, etc. One instrument can tell what type of blood cell is present by size. Other instrument can measure the different characteristics of the cell to categorize them. You can use the hemocytometer to count. This is for manual blood count. Contain counting chamber that hold a specified volume of diluted blood are used. The normal values in a CBC for male is around 4.5 to 6 million per millimeter cube, while female is a little bit lower. This number decreased with anemia and increased with erythrocytotic states such as erythrocytosis or of chronic hypoxia, dehydration, stress, polycythemia, and thalassemia minor. For procedures, it's only for knowledge, for you to know, not to study, for all the coming procedures. For counting, you first count in the 5 red cells in the square number 5. You can see from the first figure. The red cells is square in the middle, in 4 square at corners and another one in the middle. The top left touching, you count it using, you can use a counter hand tally. The red cell, actually the equation is the red cell number per microliter equal the total cell count in 5 square times 10 to the power times 10 Saturn. You can see that you only count, from the second figure, you only count the cells that's touching the top and left border. The cells touching the right and the bottom border are not counted, and this is the direction of counting. RBC disorder. The high number of red blood cells may indicate some of the conditions, like low oxygen tension, like congenital heart diseases, like core pulmonale, which is acute strain or hypertrophy of the right ventricle caused by disorder of the lung or of the pulmonary blood vessels. It can indicate pulmonary fibrosis, polycythemia, dehydration, renal or kidney failure, renal tumor, high erythropoietin production. The low number of red blood cells indicate blood loss, anemia, hemorrhage, bone marrow failure, erythropoietin deficiency, hemolysis from transfusion reactions, leukemia, a lot of types of leukemia, multiple myeloma, which is a malignant proliferation of plasma cells in bone marrow using numerous tumors and characterizing by presence of abnormal protein in the blood, malnutrition, defect in iron absorption, decrease in hemoglobin causing rupture of RBCs, and can indicate as well the low number of RBCs over hydration. The blood diseases involving the red blood cells include anemias, all types of anemias. Iron deficiency anemia is the most common anemia. It occurs when the dietary intake or absorption of iron is insufficient and hemoglobin, which contains iron, cannot be formed. Sickle cell disease, and of course all of you are familiar with the sickle cell disease. Thalassemia, it's a genetic disease that results in the production of an abnormal ratio of hemoglobin subunits. Spherocytosis, it's a genetic disease that causes a defect in red blood cells cytoskeleton, causing the red blood cells to be smaller in size, spherical in shape, and very fragile instead of donut shape and flexible. Anemias, continue this slide, anemias as well. They have a pernicious anemia, which is an autoimmune disease where the body lacks the factor required to absorb vitamin B12 from food, which is needed actually in the making of and production of hemoglobin. Aplastic anemia caused by the inability of the bone marrow to produce blood cells. You have pure red cells. Aplasia is caused by the inability of the bone marrow to produce only red blood cells. Hemolysis, of course, you know, hemolysis is the excessive breakdown of red blood cells. The malaria parasite spends the blood count can reflect if there is a malaria parasite, because malaria spends part of its life inside blood cells, feed on their hemoglobin, and then break them apart, causing fever. Polycythemia or erythrocytosis disease characterized by excessive number of red blood cells, and this increases the viscosity of blood and can cause a number of symptoms. In polycythemia vera, the increased number of red cells results from an abnormality in the bone marrow.